calcium chloride, if you remember from chemistry, an ionic compound comes apart in water, creates calcium positive ions and chlorine negative ions. Does that ring a bell? When we grow the bacteria in the incubator, it's at 37.5, our body temperature, coincidentally. The bacteria like most. So they get this shock for 90 seconds, and then we put them right back on ice for 2 minutes. And there's different theories about why this works, or different hypotheses about why this works. But one of the hypotheses is that it kind of opens pores on the walls or on the membrane of the bacteria, kind of like you would sit in a sauna and have it open your pores. And your ice can get dumped down the sink. So everybody should have it in a little tube like this. Okay? And then we're going to put it in the incubator over there. That's good.
So some of the steps that we took to ensure that the plasmid would get inside the bacteria is what we did was we put the bacteria and the plasmid in a solution of calcium chloride, and since that was ionic, it meant that there would be positive and negative parts, and so since DNA is negative, it would stick to certain parts of the bacteria. So then what we did was we put it in this heat. We put it in water that was really, really hot so that we could heat it up, and essentially the pores on the bacteria would open up. And so some of the plasmids would fit inside there, and so some of the bacteria would get the protein and they would be able to glow.
I'm thinking about this. I'm thinking, what's the logical order? Because I'm only going to give you 2 of these. I'm going to give you one spreader that you should be in contact with our control, and one spreader in contact with the plasmid. So what's the logical order? Yeah. One with antibiotic on it or one without antibiotic?
Without.
Yeah, so do this one first. Does that make sense?
Marta wants to let me.
You guys move these petri dishes really slowly once you put the bacteria on board. Do your best.
You look like you're having fun.
I think you guys all saw this, that we really have good satellite colonies that we talked about in this one. It's like perfect example of it. Also in is it lemon? Very nice.
Not so good. It's okay.
No, it's pretty good. So remember that the transformed ones are the ones that show color. The satellites are not transformed, so we don't want those. They're not going to be paint for us in our experiment. So in order to make paint, we need to get a little bit of those transformed ones and have them grow up into, to proliferate. We need a whole lot of them.